All right, so our project is about reactivity-informed design and synthesis of andrographolide and analogs NF-kappa B modulators for cancer treatment and degenerative diseases. So our lead compound is andrographolide, which is a natural product diterpenoid extracted from Andrographis paniculata, which is a plant found in South India and Sri Lanka. As a self-preservation mechanism, Andrographis paniculata tastes bitter to the animals that try to eat it, andrographolide being the reason behind why the plant is also known as the king of bitters. Beyond just being bitter, andrographolide is currently also in clinical trials for multiple sclerosis, HIV and AIDS, and has anti-cancer properties. Andrographolide has been found to exert its biological property through the inhibition of NF-kappa B, which is a transcription factor at the crossroads of a myriad of cell signaling pathways, including those pertaining to tumor survival and metastasis. NF-kappa B is a heterodimer of a P50 and P65 subunit. And recent papers have also found that andrographolide exhibits activity in antimicrobial, antidepressant, and anti-inflammatory ways. So andrographolide has two main parts, the C-ring butenolide on the top and the AB ring trans-decalin. The C-ring butenolide drives reactivity and biological activity due to its ability to form a covalent adduct with NF-kappa B. It's also a strong Michael acceptor, which makes it an effective inhibitor for the NF-kappa B pathway. The AB ring trans-decalin, on the other hand, enables binding and alters bioactivity and allows andrographolide access to the NF-kappa B pathway. So andrographolide already works well, which brings up the question of why we would continue to further modify it. As synthetic chemists, our goal is to take molecules from nature and optimize them for human use. Fun fact, natural products comprise 3.8% of products that are used in the clinic, whereas molecules that are derived from natural products comprise 19%, which is over 5 times the number of natural products themselves. So plants don't really care about curing cancer, but we do, and this is why we chose to make analogs of andrographolide for better versions of natural products that could potentially be more potent, less toxic, and more effective. And there's also a couple of goals in developing natural product analogs, which includes improving potency, new mechanisms of actions, which is how a drug or another substance produces an effect on the body, and exploring these new mechanisms of actions by making analogs can lead to the discovery of novel therapeutics. And lastly, another goal with synthesis can be improved physical properties, which includes the manipulation of the rate of hydrolysis and enhancing stability. And as you can see on this slide, this analog was made for rapid DNA cleavage.
So now I'm just going to discuss some top and bottom piece analogs of andrographolide. So when we refer to top piece analogs, we mean the substitutions were made on the C-ring or top part of andrographolide. And when we refer to bottom piece, we're talking about substitutions on the A or B rings or the bottom of andrographolide. And this was also discussed in our Pharmacore model. Now about some of the analogs and how we synthesize them. First, we have acetonide, which is pictured right next to andrographolide, and we synthesize this compound by taking andrographolide and treating it with 2,2-dimethoxypropane with this slightly acidic catalyst. We diversified this acetonide into a number of C-ring analogs, including installation with C14 acetate, with neat acetic anhydride, and treating that with base eliminates from 1415 to give this dying product right here. Conversely, we took andrographolide or acetonide and we treated it with DBU or phenyl triphate, and we got this other dying product, which is pictured on the bottom. It's called 1112 elimination. And these compounds were all previously acid. So now about acetonide, which is a C-ring analog of andrographolide. We ran some experiments and found its partition coefficient was much higher than that of andrographolide. Now, why does this matter and what does that mean? A high partition coefficient usually just indicates towards a compound that is very hydrophobic and nonpolar, which means that it has a strong permeability of the cell membrane. This quality is really important because it's very useful for easy targeting of pathways such as NF-kappa B. Acetonide has also been literature reported as more potent than andrographolide, which really shows us the power of synthesis. It's also very important for these analogs to be something called Michael acceptors, as the ability to accept the Michael increases the effectivity to which an analog can inhibit pathways such as NF-kappa B. We wanted to see how our alterations of andrographolide impacted the Michael acceptor abilities of each of these compounds. To do this, we set up a competitive disulfide binding assay. So just a little bit about the process, we put one of the analogs into an aqueous buffer and added a substance called glutathione. Why did we do this? Because glutathione is a substance that binds to the analog, and the better Michael acceptor the analog is, the more it will bind to glutathione. So the analog and the glutathione would react, creating a new compound, and whatever the analog did not or could not react with stayed over as leftover glutathione. We checked on this reaction every 10, 20, 30 minutes to quench a part of the reaction to analyze. In the end, we were left with any amount of the glutathione that did not react with the analog. Now, as we mentioned before, the more glutathione binded and reacted to the analog, the better Michael acceptor and therefore better inhibitor it was. So based on this information, we wanted to measure how much glutathione was left for each of these compounds in order to find the best Michael acceptor. We used the leftover parts of the solution and mixed it with a substance called DTNB. When DTNB and glutathione mix, they make 5-thiol-2-nitrobenzoic acid, which is a yellow compound. We put this substance onto a plate reader, which read how yellow each of these compounds were. The more yellow it was, the more glutathione had reacted with DTNB, meaning less glutathione had binded to the actual analog. This basically just showed us that the analog was a bad Michael acceptor and a bad inhibitor. So essentially, we were looking for an analog that was that read relatively the least amount of yellow.
Now moving on, we'll be talking about some of our results from our assay. So as you can see here, the blue line represents andrographolide, the red one here represents acetonide, the yellow one here represents acetate, the green one represents the 14-15 elimination, the orange one represents the 11-12 elimination, and finally this turquoise line represents our control group. So we can tell a lot of things from this chart. So first of all, we can tell that andrographolide, acetonide, and the 14-15 elimination all had pretty similar results, which means their Michael acceptor capabilities are pretty much the same. This also tells us that even though we eliminated C14 and C15 specifically for the 14-15 elimination, it didn't have much of an effect on how good the Michael acceptor would be. The worst of all of our analogs was acetate, as you can see here, and the best analog was the 11-12 elimination, and we think this is due to a structural change, which is why it was able to be a better Michael acceptor. So now we'll be explaining why we think the 11-12 elimination worked better. So as you can see, for andrographolide, acetonide, acetate, and the 14-15 elimination, the Michael acceptor is at C12, which is right here. However, for our 11-12 elimination, since we were eliminating C11 and C12, the Michael acceptor has to bind somewhere else, which is why it's actually at C14 just for the 11-12 elimination. Now going back to andrographolide, we can see here at C17, there is this olefin. And we believe that this olefin at C17 is blocking C12, which is why the Michael acceptor wasn't able to do as well. However, for our 11-12 elimination, since the Michael acceptor was on the other side, there was no reason for the C17 olefin to block it, which is why it performed much better than the other analogs. We're currently testing our theory by deleting the olefin to see if it has any changes in our results. Some future work that we're currently wanting to do is we're trying to make C17 more sterically encumbered, and we're going to do this by adding different substitutions such as cyclopropanes and epoxy groups. We're also trying to improve the metabolic stability of the compound by tying the diol of the AB ring to other groups, and we're testing multiple C-ring substituents to see which one would be the best Michael acceptor. We'd like to give some recognition and appreciation. We would like to thank our advisor, Edward New. We'd also like to thank the ASDRP team and lab staff. And finally, we'd like to thank the rest of the andrographolide subgroup for all of their hard work.
Thank you for listening. Please let us know if you have any questions.